We're going to get started in about 1 minute, just letting the attendees continue to join. Okay, good evening, everybody. On behalf of the IgA Foundation, I'd like to welcome everybody to our first webinar of 2024 with Vera Therapeutics, one of our clinical trials that is happening specifically for IgA nephropathy. This evening, we have Dr. Brenner, Al, and Marshall from Vera, who's going to give our webinar this evening. Questions can be typed in the chat box if you have questions that will be answered towards the end. I'm going to turn this over to Marshall and the team at Vera. Thank you.
John, thanks so much. And thank you to everyone tonight for joining this webinar. I know that the IgA Nephropathy Foundation pulls together an incredible community of people, patients, family, friends, who are interested in making sure that they have a healthy life for themselves and their loved ones around the diagnosis of IgA nephropathy. My name is Marshall Fordyce. I'm a physician and the founder and CEO of Vera Therapeutics, and it's just been a great pleasure to work with John Marsala and the entire IgA Nephropathy Foundation family. And I can say that after being engaged with you all for the last several years, I want to specifically thank Lenny, Brian, John, Margaret and Michael Warner, and especially Bonnie and Stuart on the operating side. We've been focused on developing a new treatment for IgA nephropathy, and this is pretty great timing because we just shared exciting new data from our phase 2 trial, and we are now enrolling our phase 3 trial. So fantastic timing to work with the foundation to let you know about the new treatment that we're developing, what we see as evidence of its safety and how it may work. And we think this may, in fact, really raise the bar for what patients could look forward to and what physicians can provide to their patients. So without further ado, let me go to the next slide, Al. I want to tell you who we are. Vera Therapeutics is a company based in the Bay Area. This is a good number of us at Kidney Day holding our fists down by our lumbar regions. This is us showing our kidneys on CKD Kidney Walk Day. And important that you all know as we share the science and clinical medicine of tonight that Vera is made up of people who are committed to making a difference in the lives of IgAN patients. And I think it's no surprise as you get to a company of about our size, there are many people who are affected by kidney disease, and we actually have several kidney transplant donors within the Vera team. So this is a group driven by passion and really guided by science. And in my view, from the very beginning, our best hope, medicine's best hope, is rigorous science, and science that turns out to show safety and efficacy in the long term. And it's an incredibly exciting moment in this field because we are making advances. And I want to give Dr. Rob Brenner, our Chief Medical Officer, an opportunity to share some of that with you tonight. Dr. Rob Brenner is Vera's Chief Medical Officer. He is a physician and nephrologist of over 25 years, and has been both a clinician and a researcher during that time. He trained in internal medicine at the Brigham and Women's Hospital at Harvard in Boston, and then did his fellowship in nephrology at Stanford University with Dr. Richard Lafayette, who some of you may know as an important opinion leader in the field. With this, I'd like to hand it to you, Rob. Thanks so much for your leadership and your passion for our work at Vera.
Great. Thank you, Marshall. And it really is a pleasure and an honor to be able to address all of you. And I couldn't be more enthusiastic about the opportunity to tell you about our lead drug, atacicept, which all of my colleagues at Vera are hard at work doing everything we can to bring this drug to patients in the future. So atacicept represents a real, I would say, disruptive possibility for patients with IgA nephropathy, and that is to have the opportunity to disrupt the disease and to produce what we would call a disease-modifying result for patients. And that's because we understand how IgA nephropathy progresses. We understand the underlying aspects of the disease, what's driving it, and that has allowed the generation of this drug, which really acts specifically to change outcomes for patients, and that's what we'll go through through this communication. So we'll get into the details, but importantly for patients is to recognize that this is kind of a precision therapy for IgA nephropathy, and it's administered by patients themselves as a self-injection, and in the phase 2 and now the phase 3 program. So in the clinical trials that will lead to eventual approval, it's injected once weekly with a very small needle, which has been very well tolerated by the patients in the clinical program. And we have an aggregate, a safety data set in more than 1,500 individuals from a variety of clinical trials across many different diseases, all of which defends a more than acceptable safety profile. And as we'll talk about now, the trials in IgA nephropathy specifically have really not only met, but exceeded our expectations about what the promise will be in the future for patients. Next slide. So I think one of the things that gives us the confidence that we do about the drug is because we understand that IgA nephropathy is a disease that begins with, let's say, alternative function of B cells, a cell population that's part of the immune system. And in this alternative function, there is the generation of what we call immune complexes, which get stuck in the kidney. They get trapped in the filtering component of the kidney. And if we begin on the left, we see two words, BAFF and APRIL. Think of BAFF and APRIL, we call them cytokines, but these are proteins that circulate in the body. And I think of BAFF and APRIL as the two energy sources for B cells. The same way a hybrid car can use both gasoline and electricity to move the vehicle forward. Both BAFF and APRIL are the two components that drive the activity of B cells. And we know that in IgA nephropathy, the levels of BAFF and APRIL can go up. And what happens is the production from B cells, the engine of the B cells, releases a specific kind of IgA, which is where the name of the disease comes from. And this IgA, the detail is we call it galactose deficient IgA1. But what's important is that it means that the IgA doesn't have the normal amount of sugar that's coating the IgA around its periphery. So think of a Christmas tree that when it's all set up for the holidays is been adorned with lots of ornaments. And those ornaments change the way that the tree looks. If after the holidays are over and you remove all of those ornaments, you're left seeing more of the tree than you do when it's decorated. And what that allows is for the binding of another output from the B cells and plasma cells. to bind and cause a complex to form, a union of these red and yellow immunoglobulins. And those are immune complexes. They circulate, but in IgA nephropathy, they get trapped in the kidney in the filtering unit or the glomerulus, where they lead to inflammation and chronic changes that result in protein in the urine, blood in the urine, and most importantly, a decline in kidney function over time, or we describe that as a loss of GFR or glomerular filtration. So it's important to recognize that because we understand how the disease progresses and what the components are in the disease story, we can now think about a precision approach to interrupt it. Next slide. So if we were able to come up with the interruption of the disease, because we understand the mechanism, we would expect to see a quartet of findings or 4 findings. One, we would expect to see a reduction in this population of IgA that's exposed where it can be bound by other immunoglobulins. So that's the Gd-IgA1. And we know that those levels are high in IgA1. And so if we can act on the disease and turn off the faucet of production, we would expect those the amount of Gd-IgA1 to come down. If we do that and we don't form immune complexes and they don't get trapped in the kidney, we would expect to reduce the inflammation in the kidney. And a clinical consequence of the inflammation is hematuria or blood in the urine. So we would expect to see resolution of blood in the urine. 2, we wouldn't expect to see the chronic changes that occur in the filtering component of the kidney, which usually leads to protein in the urine. So we'd expect to see a reduction in the amount of protein that we can measure. And most importantly, we'd expect to see a flat picture over time for an individual's GFR, or a measure of their kidney function. So decrease the immune complexes, decrease the active inflammation as measured by hematuria, decrease more chronic changes or proteinuria, and have stable kidney function. This would be the ideal picture if we had a true disease-modifying approach to this disease. Next slide. And here are the data that my colleagues and I shared with the community at the end of January. And this is the quartet of measures that I had just shown idealized. And now we've got the real world data from our 72-week or a year and a half follow-up from our phase 2 trial. On the left, we see that there was about a 2/3 reduction in the amount of this Gd-IgA1 driving immune complex formation. 2, we saw that over 80% of patients who had blood in their urine when they entered the trial had resolution of blood in their urine while on therapy. 3, we saw about a 50% reduction in the amount of protein in the urine in these patients, a dramatic reduction, and a reduction that is well in excess of what the US FDA has used for initial approval for other products in the space. And most importantly, on the far right side, we see that the GFR that patients entered with at the beginning of the trial was identical to what the GFR was after a year and a half. As a nephrologist, I can't tell you how exciting that flat line is. And in many ways, it is distinct from a lot of the drugs that have been approved over the years for different forms of kidney disease, where the best we can do is slow the progression, but we can't halt the disease. And conceptually, the way I think about this is patients entered the trial exhibiting all the signs and the symptoms and the laboratory evidence of having progressive IgA nephropathy. But after 72 weeks or a year and a half on atacicept, we have now transferred them from this picture of having IgA nephropathy and progressive kidney disease to one where their kidney function over time now looks like the profile of the general population, not of patients. This was a monumental moment, I think, for clinical trials in IgA nephropathy, and it certainly was an important moment for everyone involved, the patients, the investigators, and us as the sponsor, and gives us great confidence that with a confirmatory phase 3 trial, we will be in a position to make this drug available around the world for patients who are in need. Next slide. So I want to dive a little bit further into the statement I just made about how to think about the GFR or the kidney function profile over time in the general population. So in this figure, if you see there's a black line at 0 that goes from left to right. And this shows over time what a stable, unchanging measure would be. And what we have in the light blue line or the gray line is an estimate from the general population. And if we look at a variety of different sources, we can see that in general, the population of healthy people loses about 1 mil per minute in their GFR a year. So a very small reduction, but a steady reduction as we age. Next, we see what happens to patients who have a diagnosis of IgA nephropathy and have been treated with supportive therapies, but without a precision intervention that turns off the faucet of the disease process. And these patients are losing GFR at a much more rapid rate, which is why over time, patients with IgA nephropathy often require dialysis or transplantation because they have progressed to overt kidney failure. Next. Now we've superimposed the results from the atacicept 1.5-year data follow-up, where we see that the GFR profile over time, 1 is stable, and 2 looks much more like that of the general population than a group of patients who have been diagnosed with IgA nephropathy and are progressing at a steady rate. This is really what offers the great promise for the future for us to have an opportunity to make available to patients a drug that changes their profile from one of having a progressive kidney disease to one where we've turned off the faucet of the disease process and allowed patients to not have the concern about a further loss of their kidney function. Next slide. So to summarize, this phase 2 trial met its primary endpoint where we reduced proteinuria versus placebo after 9 months. And now after 18 months, we have this consistent picture of having benefit across the quartet of parameters that we're measuring. Gd-IgA1 goes down, hematuria resolves, proteinuria is cut by about 50%, and GFR is stable. Importantly, the safety profile in both the first 9 months when we had a placebo control and in the open label extension, the safety profile was comparable to placebo in the first 9 months and consistent with what we would expect to see in a population that has IgA nephropathy. Next slide. So now we can talk a little bit about the phase 3 trial, which is actively recruiting. And I think it's important to remember that to get a drug approved in the US and in other parts of the world, we go through 3 stages of clinical trials. We do original phase 1 experiments, often in healthy people, to see if there's any aggressive safety reason that would cause us to stop looking at the drug in a population who has a disease. And then in phase 2 trials, we do 1 or 2 studies sequentially to help determine what the right dose is, how to administer the drug, and get a preliminary view of the safety and the clinical benefits. And then in phase 3, we do a larger confirmatory trial that builds off the experience that was generated in phase 2, and the phase 3 data are the key data that are used when we file with regulatory authorities for approval. Now, to the credit of the Vera team, they engineered a phase 2 program that was bigger and longer and more substantive than usual. And the way that the drug was administered in the phase 2 is identical to the way it's administered in phase 3. In aggregate, the breadth of the phase 2 program and the design of the phase 2 relative to the phase 3 gives us a lot of confidence that we will see in the phase 3 results very similar findings that we've seen thus far in the phase 2 program. Next slide. So who can join the study? Well, participants will complete a variety of health assessments to find out if they are eligible to join this particular trial. They need to be at least 18 years old, but there's no upper age limit barrier. They need to have had a biopsy that reveals IgA nephropathy, and that has to have occurred within the past 10 years, which I think will be a very low barrier for entry. And then if they're using other drugs that are generally good for patients with chronic kidney disease, but are not specific to IgA nephropathy, drugs like ACE inhibitors or angiotensin receptor blockers or ARBs, or even newer drugs like SGLT2 inhibitors, they just need to be on stable doses of those drugs. The reason for that is those drugs can have an impact on the amount of protein in the urine. And so because we want to look at the impact of atacicept versus placebo, it's important that if participants are on those drugs, that the dose of those drugs shouldn't be changing so that we get a clearer view of the impact of atacicept. There are a variety of other requirements that each study clinic will evaluate and ultimately will determine the eligibility of an individual participant to enroll. Next slide. What can the participants in the trial expect? Well, the trial itself is long because some of the follow-up is quite long. It could take up to 3.5 years in total. Participants will be supported in terms of their travel and their time, and the intervention will be a once-weekly injection of either atacicept or placebo, just the way we did it in the phase 2 trial. We know that patients were able to tolerate this very well, and we did not have patients dropping out over the 1.5-year follow-up to date because they found the dosing and administration of self-injection to be overly burdensome. Patients will be monitored by the clinical team at the study site, and we will use a mobile app system or a website tool to help ensure that we are tracking the injections that patients are receiving. There will be ample opportunity for the participants to learn how to self-administer the injection themselves. It's something that worked very well in the phase 2 trial, and I'm confident based on our aggregate experience about using self-administered weekly injections for many medicines in healthcare today, that it's something that all participants should look at as something that will be a very easy thing to get accustomed to. And then participants will receive all of the study supplies that are needed to complete the trial, all the tools, all the things that will be used as part of the investigation. Next slide. So here is a diagram that simply relays how the trial will be executed. In the beginning, there'll be a screening period where the suitability of the participant for enrollment will be determined. That takes about a month, and there'll be a physical examination and blood tests and urine tests to make sure that patients qualify. Then, if they're eligible, patients will get randomized. That means half the patients will get randomized to receive placebo, half will get randomized to receive atacicept. The patients won't know what they were randomized to, and the sites won't know what they were randomized to, and the Vera team won't know what they were randomized to. It will all be blinded, and patients will just be on study material. but no one will know if they're getting placebo or atacicept. That allows us to really glean what we need to from the results, that it's a true double-blind experience. The treatment will continue for 2 years' time with weekly self-administered injections and intermittent follow-up with the clinical team and regular assessments for a variety of parameters over those 2 years. And then after those 2 years, for all those subjects who completed, we will offer an open label extension, just as we did in the phase 2 program, where everyone, regardless of whether they were on placebo or atacicept, will now move to atacicept, and that will continue for at least another year. And we will continue to follow those patients and collect their long-term data to give us even more information about how the drug is behaving over a very extended period of time. And there'll be some follow-up when the trial comes to conclusion. Next slide. So now we can get into some questions and answers. We have some questions that were previously submitted that I'm happy to go through, and we can start with that. And then John and Al, we're certainly happy to field other questions as we complete those responses. If there's anything pressing, we can handle that right now. Otherwise, I know there are some questions on the next slide.
There are some questions that just popped up here. I'll let Al read them out. I put them in our chat
Okay.
that are pertinent to the slides that just came up.
Okay, great.
Okay. So just reading questions that were just submitted, and also thank you to the audience that pre-submitted questions. We will get to those as well. The first one is Rob or Marshall, if you could speak to data on how atacicept reduced proteinuria based on baseline proteinuria levels. If not from phase 2, will this be analyzed in phase 3?
Well, you know, the totality of the data that we've seen is after a year and a half, there was nearly a 50% reduction in proteinuria from baseline and for patients in the program. So that's across all of their different baseline proteinuria levels. And the data that I've seen shows that the percent reduction is pretty consistent, irregardless of where patients were starting out.
Thank you. This is a quick one. Is there a minimum proteinuria level to participate in the phase 3 trial?
There is, and I don't know if we have a slide on that.
It should be 1 gram per day.
Yeah, it'd just be nice if we had it for people to see. But one of the things that we can certainly make available are the entry criteria, so patients have access to that. And we can certainly direct people to a web page that has all of that information.
Thank you, and this might be a good time to insert for the audience. There is a website called theoriginIgANstudy.com for more information, and you can also click to see if you are eligible for the study. There is a short survey that one can take to access that and also learn more. So it's called theoriginIgANstudy.com. A lot of interest in this trial. There's a question here about are only people in the United States eligible? Are patients from other countries eligible as well?
It's a great question. So IgA nephropathy is a global disease, and there are some populations where the prevalence or the number of people with the disease is overrepresented. It's very high prevalence in Asia, for example. Because of that, and because of our interest in examining the drug thoroughly and globally, this is a global trial. The trial is being conducted in many countries across, lots of continents. I was just at an investigator meeting a few days ago in Europe that had investigators from South America and Europe. We've got sites in Asia and lots of sites here in the United States.
Thank you. Another inclusion question. What eGFR level is too low to join the study?
Yeah, in the phase 2 program and in the phase 3, we have not allowed patients with GFRs below 30 mils per minute to enroll. the mean GFR was about 65 or so in the phase 2 program. I expect that it'll probably be similar to that, which means there'll be a broad distribution from the 30s, 40s, 50s, 60s, 70s, 80s, and probably even some patient with GFRs close to normal in the 90s. So we'll see. But it's a very broad GFR range. And I think what the other thing is, we don't exclude people, we don't censure them from the trial if they start with a GFR higher than 30, but during the course of the trial, their GFR drops below 30, they would continue on the trial. So there'll be a very broad GFR range. And so I think for many patients who are participating in this webinar, that their GFR will probably be in a zone where it's appropriate for the study.
Thank you. We have a mechanism question. The question is, how is atacicept different from steroid therapies?
It's a great question. So steroids are, I would say, blunt instruments that we use when there is a big inflammatory process that requires, let's say the big guns that have lots of unintended consequences that are not advantageous for patients. But steroids are not a specific intervention for the immune complex formation that drives IgA nephropathy. They're a tool when someone has very, very bad active nephritis or active inflammation in the kidney, or if they have lots of protein in the urine. But for someone who is early in the disease, or doesn't have a lot of blood in the urine or a lot of protein in the urine, but they have progression and their GFR is declining, steroids are not very helpful. What we really want to do is turn off the production of these immune complexes and avoid a lot of blood in the urine, avoid a lot of protein in the urine, and avoid a loss of GFR and kidney function. And so the mechanism for atacicept, by modulating the B cell activity, decreasing the Gd-IgA1, decreasing these pathologic immune complexes, is acting in a completely different way than steroids. And now, speaking as a nephrologist, if I project towards the future, my own personal opinion is, with the availability of a drug like atacicept, I think it will be very, very rare that any patients with IgA nephropathy would need steroid treatment again.
Related to this question is a question about what level of immune suppression is associated with atacicept, and I'll tag on multiple questions asking about side effects or the overall safety profile of atacicept.
Yeah, so the first thing is, as opposed to steroids, which are immunosuppressive drugs by design, they're blunt and they're broad. Atacicept is not an immunosuppressant. Atacicept is a drug that binds two cytokines, BAFF and APRIL. And BAFF and APRIL are the drivers, they're the engine source for B cell activity. So all we're doing is modulating the engine speed of B cells. We're not turning off B cells. They're still producing immunoglobulins like they normally do. But what we've done is turn down this hyper engine function that's churning out Gd-IgA1 and reducing it back down to a level that doesn't lead to immune complex formation. So importantly, we haven't seen evidence of immunosuppression. In fact, the phase 2 Origin trial was done during the COVID era. We didn't see increased evidence of COVID infection, and we didn't see an inability for those patients to mount an immune response when they were immunized against COVID, all of which I think exemplifies that atacicept isn't an immune suppressant. It's a specific precision B cell modulator. And there's a big difference between those two concepts. So we shouldn't be thinking about atacicept as another in a class of immunosuppressants. Second, the data from the placebo-controlled portion of the phase 2 trial, when we're comparing active drug and placebo didn't show a difference in infectious complications. So the overall safety profile was comparable between placebo and atacicept, and it was consistent with a background IgAN population safety profile or adverse event rate. And I think most importantly, we're not seeing differences in infectious risk, which I think is an important component of this integrated story. Again, we're talking about a drug that's modulating the engine speed of the B cell. It's not turning off B cell activity altogether.
Thank you. There is another question. Again, we have a lot of great eligibility questions. Would suggest that the audience please go to theoriginIgANstudy.com where you can get detailed information about your eligibility. But if maybe just briefly, if there are patients interested who are currently on a endothelin antagonist or a steroid, are they eligible for the trial?
Yeah, they're not eligible for the trial if they're actively receiving a steroid. But if they've received steroids in the past, I believe they're eligible. The endothelin receptor antagonist is a little bit of a more difficult one. When the trial was designed, these drugs were really just becoming available, and they only really would have been used in experimental situations before that. And for that reason, it was written that if patients had received an endothelin receptor antagonist, that they would not be eligible, even if they were no longer on it. It is something that we'll pay attention to, but at the moment, that would be an exclusion criteria.
Thank you. Can atacicept be used in conjunction with other treatments?
It not only can be used in conjunction with other treatments, it has been used in conjunction with other treatments. And now, speaking with my clinical nephrologist hat on, my expectation is that it will be used in the future with other agents. The key is which other agents. So I look at atacicept as the precision intervention for IgA nephropathy that's turning off the faucet of immune complex generation. But there are other drugs that we use generally that I'm sure almost every patient who's on this webinar right now is using. Those are ACE inhibitors or angiotensin receptor blockers, so-called RAS agents that are good for kidney health for patients who have chronic kidney disease. And more patients are also using SGLT2 inhibitors, which, as we've learned in recent years, are relevant for patients, again, with general chronic kidney disease. If I was treating a family member or a patient who had IgA nephropathy in a world where atacicept was available, I would use that first for a specific IgA nephropathy benefit, but I would also prescribe an ACE or an ARB agent and maybe an SGLT2 for overall CKD therapy. And that, to me, would be the ideal cocktail. In the Origin phase 2 program, almost all patients were on an ACE or an ARB therapy when they enrolled. They were on a stable dose. And there were many patients who were also on SGLT2 inhibitors. In Origin 3, we're welcoming patients who are on stable doses of ACE inhibitors or ARBs, as well as SGLT2 inhibitors. So in my mind, that represents the best CKD management, but also now the opportunity for precision IgA nephropathy intervention.
Thank you very much. There is a combo question. I'll combine a couple. Can this treatment be used for preventative measures once diagnosed with IgAN? And relatedly, can it be used to prevent further kidney damage?
Well, I think the data that now everyone's seen, where we show that the kidney function over time doesn't decline, shows that the main reason to think about using it is to change the kidney outcome for someone from a picture of progressive loss of kidney function to stability. So absolutely, this drug represents something that can prevent the long-term consequences of kidney failure that are so attendant in patients who've received a diagnosis. Now, when we think about prevention, it's an interesting question. For someone to have gotten to the point where they've received a kidney biopsy means they presented in some clinical setting with some combination of the following. They've had blood in their urine on a dipstick, they've had protein in their urine on a dipstick, or they've had a blood test that shows that their GFR is low. Or they have a combination of two or all three of those. And that's led to a referral to a nephrologist. And in consultation with the patient, they've decided to go and get a piece of kidney tissue to look under a microscope to make a diagnosis. So there's already some disease process at play if someone's gone through and had a biopsy, and now we've made the diagnosis of IgA nephropathy. That said, many patients who are newly diagnosed with IgA nephropathy may have preserved kidney function in terms of their GFR. And if they're producing immune complexes, and we can see those on the biopsy, in my mind, they're a future candidate for this drug. So our clinical program is looking at patients with a certain severity of IgA nephropathy, but over time, our team will be looking at patients with, let's say, less severe disease, because we believe that if you're forming immune complexes, the most important thing to do is turn off the faucet of the generation of those immune complexes so that they don't deposit in the kidney and we don't get the kidney disease that will result. So I definitely think of this over in the future as a drug that can be used to prevent more significant kidney disease from ever occurring.
Thank you. Just cleaning up some of the open unanswered questions. If a patient has Crohn's disease, may they participate in the trial?
Marshall, you may know. I don't know if we specifically call that out. However, if they're on immunosuppressive therapy, if they're on steroids for their Crohn's disease, then that would be an exclusion criteria. I don't know if Crohn's, if it was in a steady state and they weren't on aggressive therapy, if that would be a reason for exclusion. I'm not certain.
Yeah, I think we need to look at the detail there, but there's some allowance for example, minor steroid use in the case of asthma, but more significant steroid use needs to have multiple months steroid-free before screening for the Origin 3 study.
There was one lingering one as well. You answered the low eGFR question for inclusion, but then there was a question about entry into the trial if they have already had a transplant.
Yeah, my understanding is that the trial is not designed to include transplant recipients, but it is something that we're thinking about for the future.
That question and the prevention question are so good because it reflects a good understanding of what these data tell us, and it's definitely part of our thinking for the future, but not for this current trial.
Thank you. Again, this is just a clarification. Does atacicept's MOA impact already formed Gd-IgA1 and immune complexes resulting from that?
No, what it's doing is decreasing the faucet of new production. Now, one of the things that we're interested in learning over time, since we're seeing such stable GFR, resolution of hematuria, or the blood in the urine, which means less inflammation, and reduction in the amount of protein in the urine, is I do think it's possible that if some patients would undergo sequential biopsies while they're on therapy, we might see that the biopsy picture of the magnitude of their disease might regress. Whether that's due to the body healing, the kidney healing, since there's no more or there's a lot less immune complex generation entering to the kidney in the first place, versus there is a process to kind of clear the toxic immune complexes from the glomeruli. We'll only know if we end up generating data where we've got biopsies kind of before and after. To this point, we haven't done that, but it's something that my colleagues and I are actively thinking about.
Thank you. The question about inclusion for patients on an endothelin receptor antagonist was answered. However, there's a follow-up about in the future, if this drug is approved by the FDA, can patients who are on an endothelin antagonist now go on atacicept in the future?
I think the answer to that will be yes. And I would expect over time, we will do trials that would include patients who are on stable doses of endothelin receptor antagonists, just like we're doing with ACE inhibitors or angiotensin receptor blockers. In my mind, those three classes, those three kinds of agents are all having the same general effect on the kidney. It's just that at the time that this program was designed, the endothelin receptor antagonists were still kind of investigative. And for that reason, they weren't allowed in this trial.
Thank you. This is a provocative question. Can Vera's atacicept potentially lead to a cure?
Well, I would say for those participants in the Origin 2B study who went out a year and a half, the drug basically converted them from a picture of a kidney disease to one of a profile of kidney function that is similar to the general population. Some people would call that a functional cure. John Barrett, one of our lead advisors and one of the major experts in IgA nephropathy, called this a functional cure when he saw the data. And when I go to talk about that, some of the earliest data that he saw back when Marshall made the bold decision to bring this drug into Vera for us to develop. And I think that the vision of, or the promise of what it looked like, years ago on a very small data set, is now something that we have demonstrated in this large phase 2 trial. But certainly that's the way that my colleagues and I are thinking about it.
Thank you. There's a question I can answer. There is a patient ex-US who is trying to sign up on the website. And if you're having technical issues because you don't have a US phone number, there is a contact us button where you can see an email address. So please use that and our team will triage that for you. Thank you. There's another question, and we can answer based on what the company has disclosed publicly. But when does the company anticipate FDA approval if all aspects go as planned?
Marshall, you want to take that one?
Sure. According to our current projection, we are anticipating approval in 2026.
Great. Okay, and thank you everybody for your questions. We just have a couple left here. Does the dosage change with a patient's weight?
No.
Okay, and the last one that we see that is unanswered so far is it true that Origin 3 is going to be produced to start treatment in Japan beginning in 3 months? Not following that, if the person who submitted can re-clarify that question, or if Rob or Marshall, if you understand how to answer that.
I'm happy to take the question. Origin 3 is being conducted, as Dr. Brenner said, globally, and we do have multiple sites in Japan, and they are opening as we speak. So there should be an opportunity for someone interested to join in Japan to do so, if not now, soon.
Yeah, my understanding is it's happening right now.
Yeah.
Okay. This one, I'm not sure if we can answer, but it's the question is, how much will this medicine cost weekly?
I can take that question. Those are decisions that are not made at this point. We need to see all of the evidence that comes out of phase 3. And so we don't have an answer to that at this stage. That really comes at the moment of approval, when we can look at the totality of the data under review by FDA. It's an important interaction between ourselves, FDA, and the payers.
Yeah, I would say it's important. Vera is a small company, right? We're 50 some odd employees, and we have just completed these 72-week analyses, and now we're underway with a global trial. You know, the vast majority of our employees are 100% focused on helping to get patients enrolled, ensuring that patients are cared for appropriately, thinking about how we talk to the regional health authorities. And although Al is a member of our commercial team, he's isolated. We don't have a lot of people on the commercial side. We're really focused at this stage on clinical development and doing everything we can to move this program forward so we can bring this new therapy to patients.
Marshall, can I just jump in here with one question also, just to clarify that? What is their cost to be in the trial?
Cost to patient there. Rob, do you have an answer? I don't think there is one.
There's no financial cost. What we are grateful for is that patients are willing to provide informed consent to participate in this trial. It's a big commitment, and we take that commitment that a patient is willing to give to heart. We're grateful for it. We can't get the drug approved without people participating in clinical research. And so we think that the patients who are signing up, who meet eligibility and are willing to go through the totality of the program, we think they're heroes, right? And it's to their credit that we have the data that we have from the phase 2 program. And if we get the drug approved, or when we get it approved, it's going to be in large part because of the dedication of patients who said, I want to raise my hand, I want to participate in a trial, I want to help my fellow patients.
Thank you. We had 2 related questions about geography, about sites in Canada and the southeastern United States. The answer is yes, there are sites there, and to please again check the IgANstudy.com for more information there. There's a question back on the label. If this product is granted accelerated approval, is it likely to have a black box warning?
Rob?
One, I don't think from a regulatory perspective, I don't think whether a drug is approved at accelerated or for full approval, initially, there is an increased or a decreased likelihood of a black box. A black box warning is something that at least the US FDA uses if there's important safety information that is relevant for how the drug is prescribed and marketed and what patients are communicated when they start the drug. My own sense is based on what I've seen so far from the data package. I'm not expecting that these data are consistent with the need for a black box warning. So that's, until we have the final results, we don't know. And we haven't had a final conversations with the FDA. We're still, doing the trial. But I think, we're confident in the safety profile, and we're confident that Origin 3 is a very similar design, identical dosing that was used in Origin 2, and that gives us great confidence in what the results will be when the trial completes.
Thank you so much. It looks like the last one is a follow-up from the patient with Crohn's disease, just asking if their therapy is a biologic, not a steroid. Does that qualify them to be in the trial?
Yeah, I would say for an individual, the best thing to do would be to connect with members of our study team and look at that. Specifically, it's hard to know without all the details on the individual case, and I wouldn't want to give too much confidence or give someone less hope that they might be eligible. But we can dig into that in an individual basis.
Great. John, I think those were all the open questions I saw. I'll turn it back to you.
Those are all the open questions that I see as well. If you guys would like to just leave a closing remark before we do close it, that would be great.
Yeah, maybe I'll start, then Marshall, you complete. First of all, I want to really thank everyone for their participation. It's truly an honor for me as a clinician to have an audience with so many people who are motivated to stay abreast of new developments in IgA nephropathy, I couldn't be more supportive of what the foundation's work is all about. And to just let you know that there's a team of people at Vera that is 100% committed to fulfilling the promise of what these early data show us. And we couldn't be more optimistic about what the future prescription for IgA nephropathy will mean for patients around the world.
Rob, thank you so much for your presentation tonight, and fantastic questions from everybody who joined the call. Thanks for taking time in your evening or morning, wherever you are in the world tonight. I want to echo Dr. Brenner's comment. We are committed. I've been going to the IgAN Foundation Spark Conference for the last 2 summers. I bring those stories back to the headquarters with me about seeing some of you finding new connection coming out of isolation, focusing on health and diet, finding your way to new treatments. It's absolutely awe-inspiring for all of us. It motivates us. It's why we're doing what we do. If there are any questions that came up that you either didn't ask or didn't have fully answered, please let us know. And we've got a full team within the details of the study or generally. And hopefully what we've communicated is our measured hope. We do not want to get ahead of ourselves, but we're extremely excited about the phase 2 data. This phase 3 study will answer key questions and get us ever closer to what we think is going to be an exciting new option for patients. Once again, John Marsala, Bonnie, I think I saw you out there on the list, and all of the IgAN Foundation. Thank you for partnering with us. Advocacy is incredibly important. We want to support the foundation and the ambassador program and continuing to connect to this community, which I hope will grow as we bring new treatment options to patients. Thanks so much for your time tonight.
Al, would you like to add anything?
No, they said it perfectly. Thank you, everybody, for your support.
On behalf of myself, the foundation, Bonnie Schneider, and her husband Ed, with the co-founders of the foundation, we'd like to thank you, team at Vera, for all your hard work in giving the patients a dream for the future and hope for a better treatment that's out there. We thank you very much for your time this evening. Have a great night.
Thank you, John.
Thank you.